Well, so now you must be seeing the screen properly. It's okay, Inés?
Yes, we can see it.
Okay, thank you very much. So now welcome to this second session of our workshop in multi-omic data integration for human exposome studies. Well, just remind you, all the materials, slides, and even the recording from our previous session is online. In case you miss it and you couldn't watch it, you can revisualize it. It is available in YouTube. Unfortunately, it's in two parts because we had a technical interruptions, but you can freely watch it, and also you will find in our Google Drive all the materials and slides. So feel free to contact it. And here we wanted to take the opportunity also, since this is a topic related to the workshop, to announce that we are hiring in IS Global. Now we have an open position for a postdoctoral researcher in exposome research, but mainly with the goal of doing multi-omics integration analysis with the different type of data that we have here in HELIX, athlete, and other birth cohort studies. So feel free to apply if you think you can be a good candidate, and you will find all details for this position in IS Global website. So yeah, we will welcome your applications. And before giving the floor to our speakers today, I just wanted to do like a brief summary of our past session for those that they couldn't attend, or just for doing a brief recap. As you know, the different purposes of multi-omics integration pursue different objectives, no, like understanding biological mechanisms of disease, predict past exposures, or predict even future health outcomes. And this can be done with different techniques, but always we have to face different challenges. We were reviewing the different challenges of multi-omics integration, like association effects that are normally small, the need to detect mixtures or interactions, mediation analysis, also how to address or deal with the biological and technical noise or the biological interpretation of results. And we gave an overview of different approaches that are already currently available for doing that, no? Dividing them in the taxonomy, horizontal, vertical integration approaches, and how within the vertical approaches we can answer different questions like the subgroup discovery, prediction, or creating signatures or networks of biological features. And then we had the wonderful talk given by Arthur Tenenhaus presenting the framework of dimensionality reduction for multi-omics integration, and how different variations of dimensionality reduction methods can be used if we have two blocks of omics data or multiple blocks of omics data. And he presented the nice tool RGCCA with his package, RGCCA R package, and he was explaining how to use this very nice solution when we want to integrate multiple omics. So today we are gonna continue with these topics of dimensionality reduction techniques, but from a more practical dimension. And with no further ado, actually, I want to give the floor to Inès and Amine and Congron Wang. If you want, you can start sharing your screen, and yeah.
Yes. Okay. Okay. Can you see my screen?
Yeah, perfect. Thank you.
Okay. So hello, everybody. So yeah, as Augusto just said, we will do today this workshop on multi-omic dimensionality reduction techniques. So we can maybe start by introducing ourselves. So I'm Inès Amine. I'm a PhD candidate in Inserm in France. I'm supervised by Valérie Sirou. And my PhD is mainly on an athlete project on the topics of exposome, multiple health domain, and in between there's a project on multi-omic integration. Congron, I let you introduce yourself.
Thank you. Hi, everyone. My name is Congron Wang, and I'm a PhD candidate at Hasselt University in Belgium. My PhD is about the omics signatures of early life aging and use telomere length as the aging marker for my studies. And I'm also now working on a HELIX project to analyze the data of exposome and also the multi-omics.
Okay, so we can start here. So actually, Augusto just represented this slide. So we are really here in the second session of following the session of Arthur that already talked about dimensionality reduction techniques. So it was more of a theoretical session, and now it's really we will present two case study, one unsupervised and another supervised using supervised techniques. So just before, we will remind you a bit about dimension reduction techniques. So the aim of these methods is to reduce the dimension, as they say, by extracting summary variables. So a bit of vocabulary might be useful. So those summary variables, they can have several names. We can call them component, latent factors, signatures, etc. And they're actually composite variable. They're made of many variables, and to know which variable is contributing more or less in these summary variables, we have the loadings or the weights. There is two main families of techniques. There are the unsupervised technique that you might know, for example, PCA, principal component analysis, that will maximize the variance between a set of variables. So you can see it on the right. You have the principal components, and then you can see the contribution, so the loadings of each variable. And then there's also supervised techniques, which aim this time to maximize the covariance between some variables, some predictors, and a target variable. So here the real difference is that we want to predict a variable with the summary variable. So for these techniques, you have, for example, PLS, partial least square, RGCCA, which is also called multi-block PLS. But actually, here it's a tree with many multi-block dimension reduction techniques. So we won't be presenting everything today, just two of them, which are MOFA, an unsupervised method, and RGCCA, which is supervised. We also have this nice summary with some other techniques from the Mixomic paper, which is it's a package actually. And so you can see here also the unsupervised and supervised methods, and some methods can also allow for variable selection. So this can be interesting if you have a really high dimensional data. So out of the challenges that you can address with those techniques, so here Augusto presented it just before, it's actually the same that the challenges addressed by multi-omics data integration, but I think in particular it's those three. So the data heterogeneity and noise can be addressed. You can also perform mediation analysis after performing dimension reduction technique. It can help. And it's also good for biological interpretation because you will summarize some variable and you can find, for example, biological pathways. So yeah, basically this is one of the benefits of those kind of techniques. So now I will let Congron present the case studies we will present.
Yes. So today we're going to show two case studies using dimension reduction methods, and both studies are based on data from.
Hi, Congron. I think we have a problem with the microphone. Maybe you can try to switch it on and off and get solved.
Can you hear me now clearly?
Yeah.
Yes.
Yeah. Thank you. It's solved. Thank you.
Okay. Yeah, great. Thanks. So today we are showing two case studies using dimension reduction methods. And both studies are based on the data from the children of the HELIX cohorts. And here in this table, you can have a brief overview of the two studies. So in case study 1, I used an unsupervised method called the MOFA to integrate both the exposome data and multi-omics data, and then conducted a downstream analysis with the integrated data and the health outcome, which is here the BMI Z-score and binary categorization of the children, whether they are overweight or not. And in the second case study, Inès uses a supervised RGCCA model to learn the multi-omics signatures of a composite score of multiple health outcome measurements of the same children. Next slide, please. So I first introduce a bit the HELIX project. HELIX is a holistic study of exposures, molecular traits, and health phenotypes based on 6 European birth cohorts. In HELIX, early life exposome was assessed in both pregnancy and childhood using different methods and technologies. And in children, when the in childhood, when the children are aged at 6 to 11 years, molecular traits were measured in different omics platforms in urine samples or blood samples. And in addition, there are many clinical health outcomes measured at the childhood follow-up examination, including some cardiovascular, respiratory, and cognitive phenotypes. Next slide, please. In the first case study, we made use of the prenatal and postnatal exposome data from HELIX and also the omics measurements from DNA methylation, gene expression, plasma proteins, and serum and urine metabolites. To give a bit of background, in the literature, there are already many early life exposures have been associated with childhood BMI. And there are also a lot of multi-omics signatures found of groups of pollutants. And some groups of pollutants have been also identified to be related to both childhood BMI and some inflammatory biomarkers in the omics. However, no study yet has investigated whether the multi-omics can function as a mediator of the effect of the exposome on the childhood BMI. So the research question here is which are the multi-omics signatures that underlies the association between the exposome and childhood obesity or childhood BMI. The first objective of the study is to integrate the high-dimensional exposome data and the multi-omics data. And the second objective was to conduct a causal inference with mediation analysis. And this is work together with my colleagues from Hasselt University, Dr. Reimann, Dr. Alfano, Professor Plosken, and this is in collaboration with Hasselt. Next slide, please. So the dimension reduction method used in this study is the multi-omics factor analysis, or also known as MOFA. So MOFA is a factor analysis model that is used for the integration across multiple data types in an unsupervised fashion. The input for this model is multiple data matrices with measurements of various data types. You can have the likelihood of Gaussian, Bernoulli, or Poisson. And the samples in the different matrices can be the same samples or only overlapping samples, which means missing values are allowed in this model. And as the output of this model, MOFA can infer a low-dimensional set of latent factors. So that's the one you can see in a Z matrix. And these factors represent the driving sources of the variation across the different data matrices. And for the interpretation of the factors, we can use the weights or the factor loadings in the W matrices, and they show how strong a feature is correlated with a factor. And this MOFA has been implemented in both R and Python. Next slide, please. So this is a brief overview of the workflow of this study. So at the beginning, the MOFA was performed for the exposome data and the multi-omics data in parallel. For the exposome, we just simply split the data set into 6 matrices based on whether the variable, the exposure, is prenatal or postnatal, whether it is a specific external exposure or general external exposure, and whether the data type is numeric or categorical. And for the multi-omics data, because the number of omics features in DNA methylation and gene expression are much higher than in the other omics layers, so we performed a pre-filtering or pre-selection of the features, the CPGs and the transcripts, and we keep only the top 10 CPGs and the top 10 transcripts that had the highest variation. And this is because MOFA is a model that works on variance. And after the MOFA, we conducted these sequential downstream analysis. So the latent factors were extracted, but we made a further selection of the latent factors, and we want to make sure there are associations between the selected exposome factors and the multi-omics factors, and between the multi-omics factors and the outcome variables, so that we can form the triangle of the mediation. And then we used the selected factors to perform a mediation analysis. And in parallel, we also look at which specific exposures and which specific omics features that are most important in the factors. And we conducted an additional mediation analysis with those individual exposures and those omics features. Next slide, please. And now we take a look at one of the most important outputs of MOFA model, which is the percentage of the variance explained. The figures in green color are the results for the exposome MOFA model, and in blue for the multi-omics MOFA. And figure A and B show the variance explained by each factor in each data matrix, and figure C and D show the total variance explained in each data matrix that is summing up all contributions from all factors. It is notable that although the number of serum metabolites and plasma proteins are much lower than the number of CPGs and gene transcripts, even after the pre-filtering, we can see here the percentage of variance explained is the opposite. So we have much more variance explained in the serum metabolites and plasma proteins. And now with these 11 exposome factors and the 20 multi-omics factors, we fitted multiple regression models for these 2 legs of this triangle and selected only those factors that can meet in the middle for this mediation analysis. Next slide, please. So as a result of the meet in the middle selection of the factors, we obtained 7 exposome factors and 5 omics factors. And we fitted this structural equation model for each of the potential paths of the mediations. And the paths shown in black are those that are significant mediations. And if we use a cutoff value for the factor loadings to decide which are the important contributing features and exposures, we can have a closer look at the right side. So if we take the path through omics factor 6, the one that is in the blue box, if we take this as an example, we have the exposome factor 3, exposome factor 7, and multi-omics factor 6. And have a look at the right side. These 2 exposome factors seem to be a mixture of exposures from different exposure groups, and also different time windows. But the top ones can be can point to the postnatal exposures to PCBs, childhood diet, and prenatal maternal passive smoking. And for the omics factor 6, it mainly consists of gene transcripts and urine metabolites. And if we use these genes and metabolites to perform pathway analysis, we can find pathways related to antimicrobial immune response or phenylalanine metabolism. In addition to this mediation analysis, based on the latent factors, as I showed in the workflow, we also conducted mediation analysis on the individual exposures and the omics features, but I will not show it now because it's a bit beyond the topic of today. But we can still discuss afterwards if anyone is interested. So this is all I wanted to show for this case study, and Inès will continue with the second case study.
Thanks, Congron. So I think we can keep the questions for the end of the presentation. It will be easier. So for the second case study, so I will present a project where I use supervised data reduction techniques. So this time the research question, so that you have a bit of context, was to find the multi-omics signatures associated with a child health status. So it's still in HELIX, so in childhood. So here we have 3 omic layers actually measured in 2 biological layers. The first is blood DNA methylation, from which so we extracted the CPGs, the methylation level that we pre-selected a bit. And also we could extract the white blood cell composition. So we have 12 cell type proportions. And then we have the proteomics. We have 34 proteins, plasma proteins. And with these 3 blocks, we aimed at predicting the health score, which is a multi-domain health score, which has been previously built in HELIX, and that represents the child cardiometabolic, respiratory, and mental health. So it's one continuous variable. So that's an athlete project that's supervised by Valérie Sion and Léa Maître. And so you can here I put the link to a GitHub so that you can find some codes to perform the main analysis that I will show today. And you can also refer to the vignette of RGCCA because I use the RGCCA, which can be quite useful. So, so for this problem, I use the RGCCA. So it can also be referred as multi-block PLS. So the aim is to highlight latent variables from multiple blocks, multiple predicting blocks that will predict the outcome. So the optimization problem, so everything has been explained in more detail by Arthur, but just so that you remember, there's one part regarding the correlation between the latent component and the outcome. And then there's another part of the optimization problem, which is to maximize the correlation between the blocks, between the latent components of the block. And this, you choose it with the matrix C. The matrix C, so you can see if one objective is more important, if you want some blocks to be connected or not, based maybe on biological knowledge. And two important information. So all blocks have to have the same number of individuals. It doesn't allow for missing values compared to MOFA. And also an important thing is that RGCCA does not handle confounders by itself. So if you want to handle it, you can, for example, pre-correct the omic variable and the outcome variable on covariates using residualization, for example. So this is one way to do it. So the input data for this method, it's really simple. You need to give a list with the data of each block. And it's good if you can divide it in train and test so that you keep some data for the validation. And then you specify the connection matrix. So you specify which blocks you want to be connected. For this project, we connected all blocks. Then there is two hyperparameters with this model that you can choose. So the first one is the number of components for each block. And the second is the sparsity parameter, because you can perform variable selection, which can be useful if you have high dimensional data. So for us, we applied it for the DNA methylation block only. And to choose the hyperparameter, either you base it on prior knowledge, you have an hypothesis on it, or you use cross-validation, which I would recommend. There is a function which is RGCCA CV that does it, and that will produce the kind of plot you have on the right. So here, I show it for the number of components, and so you can see that the mean error for predicting the outcome is when we select 3 components for proteins, 3 for cell types, and 1 for DNA methylation. And then I don't show the plot, but we showed that the optimized sparsity parameter was 0.9, so it actually selected all CPGs here. And then you can apply the RGCCA model with this optimized parameter by calling RGCCA on the object that was created before. So what's the output of RGCCA? So you can find, it will give you the components, the biological signatures. And what's good is that you can use Bootstrap with the RGCCA Bootstrap function, which will tell you if the loadings are stable with a small variation in the data. And so it gives you a credible interval. So you know if a variable contributes significantly to the component. So for example, here I plot the first component of each block. You have one color for each block. For cell types, for example, it means that CD4 naive and Treg cells contributes significantly and negatively to the component. So the higher they are, the lower the component is. And at the opposite, eosinophil and CD4 memory, they contribute positively. So this really will help you for biological interpretation of each signature. So if I go just a bit in detail to show you an example, here it's the 2 main components that we found. It's the first white blood cell component and the first protein component. The blood cell component actually reflected immune regulation. You have a positive loading for Treg cell and low inflammation because we have low eosinophil, low neutrophil, monocyte, and eosinophiles. So this might be linked with our multi-domain health score. And then the protein component reflected pro-inflammatory status. We have low IL-1 beta, IL-6, CRP, which are linked with inflammation, low leptin, which is linked to metabolism. So this way you can get biological signatures associated with your outcome. And then the final step, you can know the performance of the model. So you can know how well does, for us, the 7 omic components predict the outcome. And this is when you will use your test set, so the 30%. So you can use RGCCA predict to predict your outcome using different models. So linear regression, but there's actually many different models. And it will give you the performance, so the root mean square error, the R square, for us, for example, was 20%, so it was really not that bad with this kind of exposome omic data. But then we had another question, which was to know if there was some component out of the 7 that explained more of the variance. And so for that, we did some complementary analysis. And so we were able to see the percentage of variance explained by each component, so the R square specific to each omic. And which really appeared was that the protein, the first protein component was the one explaining most of the variance. So it really showed that it best represents the child health status, our outcome. We showed that cell types contribute only partially. We have the first cell type component explaining 2% of the outcome variance. And what we could see was that for DNA methylation, the contribution was minimal. We had an R-square below 1%, or it might mean that the results are not replicable for an independent data. So yeah, this performance indicator really helped to have more information on the biological signatures found. So the main conclusion of this study was that we found 7 omic signatures from different layers, predicting a child multi-domain health score. So from the 3 layers, we saw that proteins best predicted the outcome with 1 inflammatory related signature. We also found 1 signature related to cell types. And we saw that in this study, DNA methylation had really minimal contribution, or it was not, the results were really not replicable. And we actually had a quite good performance for prediction with an R-square of 20%. So for us, it really helped to generate hypotheses on biological mechanism, on pathways, with a second objective that I will not show here today, which was to look at the association between those omic signatures and the exposome in order to do mediation, some kind of a mediation analysis between the exposome and health, so to see what part could be mediated by some omic pathways. So that's it for the case study. I hope it was clear. And so just to conclude, we can just state the benefits and limitation of those kind of techniques. So the dimension reduction techniques, they can allow to account for data covariance by reduce the dimension of your data. And the components that you will extract, what's good is that they are interpretable, biologically interpretable. So that can be really nice. It's also good to study multiple omic layers together. And it can help to do feature selection because you know the contribution of each biomarker. You can even do variable selection with some of the methods. But out of the limitation, we talked about the difficulty to handle confounders. It's not done by itself most of the time by the methods. The noise across omic layers, the heterogeneity, the differences in dimension. Sometimes, depending on the methods, it can lead to different results. It can be a limitation. And then there's the replication of the results. The validation or some stability tests, they are not always implemented, but I would really highly recommend that you do so because as you see, for example, in my study, DNA methylation, the results, they were not replicable. And so often you can have some overfitting issues or issues like this. So here we put again the codes, mostly in R, the 2 GitHub links. We will share with you the slides so that you can access those GitHub and here are the references. And I think that's all for us. If you have any question, we would be willing to answer them.
Thank you very much, Congron and Inès. Thank you very much for this brilliant presentation. And both case studies, I think personally, they are super high quality and illustrate very well the power of these techniques to address this integration in epidemiological settings, which is indeed a challenge itself when we have multiple cohorts as here, or even confounding effects. Also, you have shown the challenges. And I personally like a lot as well how both of you use this to, in a second stage, look for mediation, no? And these latent factors in the omics can be later used for exploring the connection with the environmental exposure, which is in the end the aim of this workshop. So very nice talk, and thank you for this work. We have indeed some questions in the chat where we are reading them. I also encourage people to use the chat if they have more questions. Here, more or less, the ones that we already have for the moment, I tried to summarize. We have some questions related to implementations detailed. One question from Yuka Hintika is regarding the MOFA. She asked about the need for scaling the data before integration, if there is a particularity in that sense, or if MOFA does it automatically.
Yeah, this scaling is done by MOFA automatically, and then MOFA will give you some for the factor loadings. You can get the non-standardized factor loadings and the standardized factor loadings based on whether the variable itself is standardized or not.
Okay, thank you. And also, continuing with MOFA, there is a question from Yaoi Zhang about the selection bias of the study population, how it can affect the conclusions in terms of how generalizable are results to other populations. For example, Inès has shown that RGCCA has this bootstrapping strategy to address this, and uncertainty is a kind of similar tool in MOFA.
Yes, I think so. In our study, we didn't do a reliability test like Inès has done, but actually we can do like fit MOFA multiple times with bootstrapped samples, and then check how many times a feature or an exposure has been selected in one factor. But at the beginning, I think we used a larger sample size, and afterwards we decided to use only the European children. And then there are indeed some difference between the results, but I think those differences are only for the subtle associations, the subtle factor loadings. But for the main driving sources of the variance, for example, the urban environment, the air pollution, those are very highly variable, and those always appeared in factor 1 of the exposome.
Oh, that's great. Well, we also have a question from Tatiana about the case study of Inès. How was the filtering performed to get the 20,000 CPGs?
Yes, that's a good question. So indeed, because the data block really had really different dimension, even if also in RGCCA there is a pre-treatment to account for the dimension, we wanted to pre-select a bit the CPGs. And so there was several steps. The first step was to pre-filter the CPGs based on technical variability. We used literature to know if some CPGs, we know that they have some measurement error, or so we could already remove them. We don't expect them actually to be truly associated with our outcome. And then we pre-selected based on the association with the exposome, because as I said, the next step is to do a mediation analysis with the exposome. So we are interested in those that are also associated with the exposome. So that's how we reduced our set of CPGs to for 50K to 20K, basically.
All right. Also for MOFA, also from Tatiana, she has a question about how to accounting for covariates. I think you already said that this is a drawback. I don't know if you did something special about confounding related to sex, batch.
Yes. For batch effects, we already residualized for the batch, or like for blood sample or urine sample collection. There are some other factors that can affect those things. We already regressed out those effects at the beginning before conducting MOFA, but there are some other covariates like sex, age, or something that we think are relevant for the biological interpretation of the omics. We keep them in not residualized. Mm-hmm. And at the stage where we try to select the factors, in the multiple regression models, we add those as covariates. And also in the mediation analysis, we add those as covariates.
Okay, that's great strategy. And also we have a question from Guadalupe. I guess that she refers to both case studies since you are using the same population, about the socioeconomic level of the different participants, and if you have checked if this affects the results or...
You want to answer? Right.
I don't know by heart, but I think it's because we have different sample sizes, but I think more or less more than 50%, higher education of the mother. Yeah.
Yeah, and I think there's maybe differences across cohorts. For example, in BIB, it's born in Bradford, it's a cohort from UK. I think there's more families from lower socioeconomic background. And yeah, for my part, I pre-corrected the variable on the covariates, so this effect should have been removed. But I don't know, Congron, if you studied it in more detail.
Yeah, we didn't regress it out at the beginning, but yeah, use it as the covariates at downstream. Yeah.
Okay. And then there is some more general questions about, also from Tatiana, about MOFA, like can be used for other purposes, since it is an unsupervised approach. Like could you use latent factors from MOFA to for example, do clustering, or like this is subtyping of individuals? Do you have an idea about it, or have you?
I haven't done it ourselves, but I think it's possible. I think now MOFA has also an extension of where you can use, in addition to different data matrices and of the samples, you can also include groups of the samples, like you have diseased or undiseased status.
Mm-hmm. Okay. And then we have, this is a challenging question. Could you, it's from David Esteban Rebellon, could you summarize what would be the advantages of using MOFA or RGCCA against other methods, like in this case PCA or PLSDA supervised? Give this example.
Well, I think one big, I don't know about MOFA, but I can say about RGCCA, compared to other methods, you can really define blocks of data. So that's for multi-omic studies, it's really a good benefits because you can include different biological layer, different dimension of data. So compared to classic PLS, that's one advantage. For MOFA, I don't know if you want to answer someone.
I think the same. We can have different data modalities in one model. Yeah.
And what's good with MOFA, if I understand well, is that it can handle also missing values, which is something
yes.
we
Absolutely.
have a lot.
This is critical. This is critical when we are integrating such heterogeneous data sources. Is a very important point. I have a questions on that, on the missing values, and also it's related to David. Next questions. Just my question is more about if there is a minimum threshold of percentage of missing values that can be handled by MOFA, like below this amount you can use it, or that is if you have more missing values than this percentage, this variable is out, or?
I think the author of this package has recommended a threshold or like the better proportion of missing values, but it doesn't impute the missing values in the data set. It uses the likelihood because when it run into a missing value, it doesn't use that missing value to update that likelihood. So there's no imputation inside. And I believe there is a recommendation of the cutoff of the proportion of missing, and maybe you can have a closer look at the paper of that package.
Yeah, but if that's the way, instead of imputing, use the available data, probably there is big recommendation about the cutoff. And also, one question about the type of data that can be included in MOFA, if it can handle categorical, numerical variables. I guess you mentioned it in your slide.
Yes, in MOFA, I'm not sure of RGCCA, but in MOFA it can handle. Now there are 3 implementations of the data type: Gaussian, Bernoulli, or Poisson. But it's always recommended to, if it's possible, it's always recommended to put your data into a numeric Gaussian format, because the other 2 types are like with approximation that might not be that accurate. And for RGCCA, maybe Inès, you can.
For my case, all my variables were continuous, but I think because I asked to Arthur one time, he said I think it's better to use numerical variables, but if you transform your categorical variables into dummies, it would work, like the package would run, and so I think it's possible as predictors, and for as predictors, but for the variable to predict, I'm not sure. I think you can also include some binary variables or categorical as dummies.
Yeah. Well, I think there are no more questions here in the chat. If I can, I wanted to comment on the fact that in both presentations, DNA methylation is a kind of residual, or is forgotten, no, in terms of variance. Explain it. Just to know your opinion about it, if you think that it could be due to the dimensionality, or if there can be other reasons for that. Do you have any hypothesis?
I think there's many hypotheses. Either it's because maybe we don't have enough individuals compared to the number of CPGs. That would be one statistical reason. Maybe it's also because in the HELIX data, it seems that the proteins are really the closest to like many phenotypes. Like Congron, she's studying BMI, and she found also the proteins. Maybe there's more variance also in the protein data than in the DNA methylation data, and those methods, they're based on variance. So if there's not that much variance, then it will not really appear. I don't know, Congron, if you have any thoughts on it.
Yeah, I agree what you said, and also because we also addressed at the end about because this kind of analysis can be confounded by the highly variable characteristics of the population. For example, the outcome variable I used, the BMI, because I have another study using telomere length as the outcome, then I don't have such a good representation of the data, because maybe the main variance inside the data is about the metabolized proteome and the BMI.
I agree with that.
Yeah.
Okay, and people, I think they are putting more questions. We still have time for going about the sample size. If there is any clue about the recommendations, like which is the minimum sample size. These are difficult questions, I guess. But
yeah, I don't know any specific cutoff. I think the good thing about data dimension reduction techniques is that it helps to solve a bit the issue of like having more variables than individuals, but yeah, no, I would not. I don't know, Congron, if you know any specific cutoff. I think 100 is minimal, but
Yeah, I think so. And it also depends on how precise your measurements are.
yeah.
For example, if you use some if you measure some methylation of the CPGs that are not that reliable, maybe those are not that precise, and you need more sample size to for the analysis.
Okay. And also there is a question from Haibing Wang about the how to select the cutoff for or the threshold for selecting relevant features in a model like MOFA. You mentioned, no, that you use this contribution or loading above 0.25, I think it was. How did you select it? It was arbitrary or?
Yes, it's kind of arbitrary and post hoc. So we first have the MOFA results, all the factors and the loadings, and we check the distribution of the loadings, and then we select the cutoff value so that we can have a good number of features left for the interpretation of the factors. So it's arbitrary. And there's no regularization as in RGCCA for MOFA. So we just use the cutoff value to select the number of features.
Okay, great. So that was a brilliant session. Also, thank you everyone for participating in the chat. It was amazing to have your questions and also to have this time for discussion. It was very nice. So thank you also the speakers for making it short. And yeah, see you everyone in the next session. It will be that time next week. Thank you very much. And yeah, I hope you enjoyed it. You will have everything recorded and all the sessions. It will be shared in the Drive on YouTube. Thank you very much.
Thank you. Thank you,
Thank
Augusto.
you.
Thank you.